So good morning. I'm Christos Shammas. I work at the Cyprus Institute of Neurology and Genetics. I will be giving you today a brief overview of the genetic insights of multiple endocrine neoplasia type 2. We will see what type of mutations are present, how they affect the functional properties of tyrosine kinase, and I'll also be showing you the molecular genetic test we actually do in our lab, and actually is a gold standard. So I believe you're getting a second introduction to this today. Takahashi and Cooper in 1985, if I'm not mistaken, first identified the RET proto-oncogene. This gene is found in the chromosome 10. Is composed of approximately 20 exons. It has a size of 55 kilodaltons and is responsible for the production of the receptor tyrosine kinase. This protein is very important, as you know. It's found on the cell wall, and it has functional properties that are responsible for the nerve cell development, spermatogenesis, kidney development, and cell differentiation and other growth factor properties. So any mutations that occur on this gene have a serious effect. This effect can be loss of function of the actual protein. Loss of function will lead to the development of Hirschsprung's disease, or gain of function. Gain of function means that this protein is working when we are not expecting it to work. Can cause MEN2 alpha, MEN2 beta, and FMTC. So MEN2 alpha and beta are considered to be autosomal dominant syndromes, which in genetics that means that just a single mutation in one of the alleles is able to produce this phenotype. Until today, there are approximately a bit more than 25 multiple endocrine neoplasia type 2 mutations identified. And I'm showing you here a small example of them. This is supposed to be the tyrosine kinase. Is found on the cell wall. Part of it is found outside the cell, and part of it is found within the cytoplasm. Outside the cell, we can find the cysteine-rich area where actually all the MEN2 alpha mutations are found, while in the tyrosine kinase domain, we can see the MEN2 beta mutations. FMTC mutations occur both in the cytosine-rich area and in the tyrosine kinase domain. So this is a poor representation of a cell. So we have a cell here, and these are supposed to be two tyrosine kinases within close proximity to each other. We have two factors here, the GDNF and the GFR1 alpha in my example. And what happens next is that these two factors, they come together and they produce the activation factor. Oops, let's go back, went too fast. So we have a signal binding site that can be identified by this activation factor. And we can see a cartoon here how these factors go and sit on that signal binding site. This does have an effect. It induces conformational changes on the actual tyrosine kinase, and as a result, they come into close proximity to each other and they form a dimer. At this stage, we can say that we have activated tyrosines formed, and this is important because, as the name suggests, kinase means an enzyme that catalyzes the transfer of phosphates. So these tyrosines now are able to accept a phosphate from adenine triphosphate. Adenine triphosphate, of course, will be reduced to adenine diphosphate. So now I'm showing you here that all tyrosines have accepted a phosphate. So we have a phosphorylated dimerization. So these phosphate groups that are present are very important because they can now activate inactive relay proteins. We can see this happening now. This activation induces conformational changes on these proteins, and as a result, gives a cell response. Okay, I'm showing you all this because it's important. It's important because mutations that occur in this tyrosine kinase actually disrupt in somehow this dimerization procedure. In the case of MEN2 alpha, when mutations are found in the cysteine-rich area, it will cause uncontrolled dimerization and uncontrolled activation of this protein. In the case of MEN2 beta, we have tyrosine kinase domain mutations, which lead to the uncontrolled dimerization of tyrosine, so it becomes active. But in this case, we have also active tyrosine monomers. And in the case of FMTC, it's interesting to know that if the mutations are found in the cysteine-rich region, they form this dimer without the presence of any activation factors, while if FMT mutations are found within the tyrosine kinase domain, they form a partial activation of the monomer. So in order to detect these mutations, we must conduct, of course, genetic analysis. Usually, genetic testing begins from whole blood, is the easiest way. We extract DNA, and then we amplify the region of interest. The region of interest in this case, of course, is the RET proto-oncogene. We amplify this region using fluorescent DNTPs, which can be then detected by Sanger sequencing, and as a result, we get an electropherogram where we can easily spot our mutation. So, with accordance to the best practice guidelines, it is important that when you request a test, it covers the above exons in your RET proto-oncogene. Usually it's done by bidirectional sequencing. This test has a clinical sensitivity of 95% for mutations that cause MEN2 alpha, 88% for FMTC, 98% for MEN2 beta. These limitations are because we are not actually sequencing the entire gene, and some rare variants actually do exist. This genetic analysis has an analytical sensitivity greater than 99%, which is actually very respectable. In cases that a patient undergoes a thyroidectomy, it's also possible to conduct genetic analysis on the actual tumoral tissue. DNA is extracted from the tissue. The genetic screening is done in the same manner as we have seen before from whole blood. If no mutation is found, no further investigation is required. But if a mutation is found from the tumoral tissue, we recommend that further analysis is conducted from whole blood. If a mutation is found in blood analysis also, then familial screening should be recommended. There are cases that we do find mutations in tumoral tissue, but not in whole blood. This means it's a somatic mutation. A lot of clinicians do not recommend any further investigation. The family is not usually screened. Okay, we have to take care of this because knowing your patient's history is very important. There are cases where patients undergo bone marrow transplant and their DNA profile in their blood system changes, and you may be missing mutations from whole blood. Here we have the stratification of the RET mutations. On your left-hand side, you can see the 20 exons. Inactivating HSCR mutations can occur in the entire gene, while activating MEN2-alpha mutations are found all in exons 10 and 11. And in MEN2-vita, the hotspot area are exons 15 and 16, of course. FMTC, as I mentioned before, mutations occur in the cysteine-rich domain and the tyrosine kinase domain also. As we have seen in the previous presentation, and I'm sure there's going to be a lot more discussion to follow, mutations, especially MEN2-alpha, have shown difference in severity. Mutations A here, this group A, are the mildest form, B, medium, and C, which is the cotton 634 mutation, which is considered very severe. The difference in severity is due to the fact that these mutations are found in a different location of the tyrosine kinase and also have a different amino acid change, which as a result has a different conformational change of the tyrosine kinase and gives a different phenotype. So, something local now. Up to now, in our laboratory, we have screened about 400 cases for the RET proto-oncogene, but in those we include family members. And so far, we have only identified one somatic case with MEN2-alpha, 8 family cases with MEN2-alpha, and one family case with MEN2-vita. And the interesting fact is that these 8 families all share the same mutation. Having feedback from our clinicians, they have told us that actually 4 families have roots that come from the same village. This village is on the mountains, which you're going to visit, I think, on Friday. This village has a population of just 65, and we thought, okay, these 4 families, since they have the same roots, they might have the same common ancestor. So what we did, we performed haplotype analysis, and we saw that they indeed have a common ancestor, these 4 families. And so we decided to do a further investigation, see, are those other 4 families also related to that ancestor? And yes, indeed they are. So this is what we call in genetics a founder effect. So Mr. Pink here most likely is a rare variant, got isolated many years ago, and many generations later, we see this variant spreading into that small community. So I'm ready to conclude with telling you that it's very important that the genetic testing analysis you request satisfies the best practice guidelines, and it is very important that these genes that are assessed actually cover, especially in the case of exon 10 and 11, are the hotspots for MEN2-alpha, and exon 16 is covered for mutations in MEN2-beta. The type of mutations can provide prognosis on the severity of the condition. You will see this again in the next presentation. Family history and family genetics testing are very important. I'm sure you already knew that. But it's also very, very important to know the genetic makeup of your population, since it will enhance your clinical investigations. And this, you need to get experience, and so you need time in order to get this type of information. Finally, I would like to thank my associates at the Cyprus Institute of Neurology and Genetics, Dr. Skordlis and Dr. Deltas at the University of Cyprus, and of course, the clinicians at the Bank of Cyprus Oncology Center for having me here and for their samples as well. Thank you.